We have an exciting session ahead, and we are starting with a very special guest. She has arrived from Missoula, Montana, so this was quite a long way, but she's here with us, Professor Anna Sala. Anna's research on tree ecophysiology and related subjects has been an inspiration to me and to others here in Israel. And she started, I think, her way with arid and semi-arid ecosystems. Now she worked mainly in more temperate regions in Montana, even boreal, I think. And for example, the work on carbon relations in trees and other plants has been seminal to the field and contributed a lot. So I'm really excited to have you here with us, Anna, and we look forward for your talk. Thank you.
So I was saying thank you very much. It is great pleasure, a privilege to be here, really. I had a long, eventful trip, but finally I'm here. So before I move on, I want to make sure to be clear that the work that I'm going to present is the result of a lot of effort from different people, collaborators, friends, students, and so my deep gratitude to all of them. I'm going to talk to you about them, give their names, but as I go. So even though I am now in Montana, I grew up in the Mediterranean, actually, and I did work in desert systems as a postdoc. And as we are aware, drought is increasing in frequency and intensity in many parts of the world. And these most recent hotter droughts have been linked to also recent tree and forest mortality events. And we care about this because forests are about 45% of the land surface area and about 75% of the terrestrial net primary productivity. Therefore, they do represent a strong, an important global carbon sink. So we do care about what happens with trees and forests in general as the climate warms. Trees under drought die because they do need a lot of water, right? They need water to, let's see if this works. I'm so afraid I will, no. They need water to expand cells when they divide and to grow. So they need water to photosynthesize and to maintain cell function, so plant living cells. Need to be under turgor pressure to be functional. And they need water because in order to acquire CO2 for photosynthesis, a lot of water is lost. And this water needs to be replaced. And the water moves from the soil to the very, very dry atmosphere following gradients of water potential. And in fact, the turgor maintenance, so the capacity to maintain living cells under turgor is really the whole goal of plant-water balance. So when we talk about plant-water relations, really we're talking about all the processes and strategies that plants use to keep turgor. And this is illustrated in this experiment that Gerard, a prior graduate student of mine, did. And what he did is he grew a bunch of ponderosa pine seedlings in small pots. He preconditioned them to drought, and then he withheld the water. And periodically during the drought, he would remove 15 of these seedlings and then rewater them to see whether they would survive or not. And then he also sampled, harvested 5 seedlings to measure different physiological. Okay, so this means that over time, we were able to build mortality curves, risk of mortality, how many seedlings would not survive as the drought proceeded relative to the physiology. And so he did that with 2 varieties of ponderosa pine, a coastal one that is less drought resistant, and an interior one that drought resistant. And the coastal one started during the days of the drought, over the drought, started dying about 2 weeks earlier relative to the Rocky Mountain. But when we express mortality, so the probability of mortality as a function of plant relative water content, and this is the relative water content weighted of the different tissues weighted by their biomass, we see that incipient mortality starts to happen at the turgor loss. So clearly indicating that sort of the importance, of course, of turgor in plants. So while plants need water, they also make carbohydrates. So this is, it's intuitive to think that water and carbon relations are going to be very much linked. And we are all familiar with the trade-off of water for CO2. So plants need to lose a lot of water in order to gain carbon for photosynthesis. Photosynthesis, the carbohydrates produced are invested either into growth, they are used for respiration and energy, they might be exported to microbes, and a fraction of it is stored. And this storage can be in the form of what we call non-structural carbohydrates, NSCs, which are starches and soluble sugars of different forms. And some plants also store fats, but I'm going to talk about the non-structural carbohydrates. And this reservoir initially, this reservoir initially was thought to be just a safeguard, a reservoir for in circumstances of stress when photosynthesis, so a source of carbon, was not enough to meet demand. So then there would be this reservoir. What happens is that these non-structural carbohydrates are really abundant. And this is a survey that we did of different growth forms in different biomes, boreal, temperate, Mediterranean, tropical, in different tissues, leaves, stems, and roots. And we see that on average, plants maintain between 8 and 14% of their biomass is non-structural carbohydrates. That's a lot. So if we have a tree, say a mature ponderosa pine tree, that's about 70 kilograms of starch and can be enough to like a couple growing seasons of carbon, right? And so this is a little puzzling, right? This could be, this amount of stored carbon could be in circumstances when it's temperature or water limit growth, cell expansion, then there's gonna be, growth is more sensitive than to temperature and to drought than photosynthesis and so in those cases plants might accumulate non-structural carbohydrates. But that in general, this is the pattern, is surprising because plants, we know, optimize carbon assimilation. We've seen that over time with the structure and biochemistry of chloroplasts, the arrangements of chloroplasts with light. This is the leaf area index in oaks where I did my dissertation. Most of the leaves are at the top of the canopy, whether we are in a valley or in a ridge top where it's riskier and they lose more water. And these oaks even change, the leaves even change the angle to optimize photosynthesis. So photosynthesis is optimized. And the question is, it is also expensive. It requires water. It requires nitrogen. And the question is, why do plants store a resource that is so costly to acquire? And we suggested that it might be because some of these stored, a large fraction, might actually be involved in water relations. And that's why there's always this amount of store. And for instance, one function could be osmotic regulation. So living cells, again, need to be, these are the living cells, need to be under turgor pressure, and yet they are supplied by water from the xylem, which is under tension. And so the question is, how come these cells don't lose all the water to the xylem? Because they are under positive pressure. And this is because they do adjust their water potential by lowering the osmotic potential. by accumulating solutes, and that offsets then the positive pressure and allows them to equilibrate or even lower their water potential. So that means that they will use solutes. These solutes can be inorganic ions, but those are not very much storable, so they are hard to store. And they are also non-structural carbohydrates, particularly soluble sugars. The starches do not have an osmotic function, but they can be hydrolyzed, so they can be a source for solutes. And these non-structural carbohydrates are also involved in stomatal behavior. They might downregulate photosynthesis if leaves are loaded with starch. And they might also be involved, like an Italian group has shown, in plant hydraulics, so in hydraulic conductance, as well as capacitance via gels that absorb water. So there is a lot that we need to learn. So then the picture of the trade-off of water for carbon can be updated to also think about the fact that plants store carbon for water. They trade carbon that cannot be invested in these other important functions precisely just to keep the water, to retain the water. So, and this vision is consistent with the fact that plants rarely deplete their non-structural carbohydrate storage and maintain very high minimums. So this is the survey that we did in about 193 species, different growth forms: conifers, evergreen, drought deciduous, winter deciduous, and herbaceous, soluble sugars, starch, and the total, and in different organs. But the summary of this picture is that these minimums as a percent of the seasonal maximum are very high, are about 36%, so close to 40%. So that means that this storage is used either on a diurnal or seasonal basis, but only up to 40% of the maximum. So this amount of storage is sort of never used, or is permanent. So that suggests that this is actually needed for immediate need signaling and perhaps osmotic regulation. So we've known forever that soluble sugars contribute to osmotic regulation, but what we do not know is how much of the storage per se, how big of a fraction is needed for that function. So that's what we didn't know. So the first question is, does storage affect water relations? This was work by an undergraduate student that was phenomenal. She grew up these seedlings, these again ponderosa pine seedlings, and kept one group in the light, and then measured different parameters over time. I'm just gonna talk about a portion of this experiment. And then she kept different groups, one of them 1 week in the dark, and then when she took them out, she measured the different parameters, the water potential, and then she put a group that was water and another group that she withheld water. She did that for another group for 2 weeks, 3 weeks, up to 6 weeks. So she had 7 groups in total with increasing exposure to dark. And I'm gonna talk about what happened here when she took the plants out of the dark and measured the water potential. A very important thing is that all these plants were water, okay? So they were not under water stress. And so what she saw is indeed a decrease of the non-structural carbohydrates. This is the average for the plant, weighted for the different tissues over time, over weeks in dark, and that's reasonable. They are not photosynthesizing, they are using the storage to respire. But what was more remarkable, and these are plants that were water, is that the water potential decrease as the plants became depleted, okay? So this depletion was despite the watering, and so this means that this effect of the non-structural carbohydrate storage is independent of soil drought, which is actually, to us, was like very puzzling. So we did more experiments. One critique that we got is that, oh, plants under dark, this is a very artificial situation. So then my graduate student at the time, Gerard, thought of a very clever way to deplete non-structural carbohydrates. So what he did is grew, again, ponderosa pine seedlings. Those are the ones that we have in the backyard. That's why we use them. He grew them in large pots, and he put 2 seedlings per pot. He sprinkled mycorrhizal spores and let them grow for a year to allow them to establish a mycorrhizal network. After a year, when we knew that there was a network formed, he shaded it, he divided the pots, and in half of them, he shaded 1 seedling. And the goal of that was that by shading the seedling now is not supplying carbon to the mycorrhizal network, and so the neighbor seedling now will have an extra carbon demand from this network. And that's how he would deplete this seedling without putting this 1 seedling in the dark, okay? So effectively, he did show a decrease of non-structural carbohydrate storage. So the seedlings in the light had highest. The seedling that was paired to the dark one was depleted relative to the light, as we expected, and the lowest was in the seedlings in the dark. And as expected, the needle osmotic potential increased from the seedlings in the light to the seedlings in the dark. And remember that an increase in the osmotic potential means a decrease of drought tolerance, right? Because this means less solutes accumulated and lower ability to lower the water potential and equilibrate with the xylem, okay? So essentially then he did see this decrease in drought tolerance. Again, these seedlings were watered as the non-structural carbohydrates were depleted. And with that, he saw that the seedlings in the dark, the black ones, lost turgor before the seedlings that were paired, the light seedlings that were paired with the dark, and these ones before the seedlings that were in the light. And this slope here is because we don't have enough points, probably it's a steeper slope, okay? So we've not done work on this yet, but there's work by the group of Craig Brotherson and Andrew McCall. What's his name? Yes, thank you. Where they have done work with vines, okay, and they had a variety that is drought sensitive and one that is less drought sensitive. And what they saw is that the more sensitive, these are cross-sections of the root, the more sensitive, as the water potential decreased, experience shrinkage of the cortex of the roots. And so when these cells lose turgor, they shrink, and then there is a loss of contact with the soil. So there is essentially cavitation between the soil and the roots. And this actually might explain that what we saw with these ponderosa pine seedlings that we did the first experiment. So their water potential was decreasing even though they were watered. How in the world could that happen? They were in the dark, they were not losing water. So what could have happened is that the roots actually shrank. And then it doesn't matter how much we water the soil, they are not getting the water, okay? Okay. So until now, we've done greenhouse experiments. I never used to do greenhouse experiments, so now I'm returning to the field, which is where nature happens, right? And so sure, we're seeing that these non-structural carbohydrates influence the water relations in the greenhouse. But does that happen in the field? Because if it does, then we have a lot of work to do in terms of modeling and whatnot. And so we're doing an experiment. We started this summer, so it's very preliminary. And it's an experiment that has different objectives. I'm gonna just present a part of this. And so we had ambient plots, we had drought plots. Guess what? Those are ponderosa pines too. And then in the plots that were ambient, or in each treatment, we had controls or lights and then shade. So we shaded one of the saplings, okay? So we had this thing for both the ambient plots and the control plots, and the idea here was we would have more non-structural carbohydrates here, and they would keep decreasing with the shade, the drought, and the drought plus the shade, okay? So just to give you a little bit of perspective, I've never worked so hard in my life, and at my point in time in my career, you should have seen me digging for the entire summer. So I'm kind of proud of this, and I'm hoping that something interesting comes out of this. But the point is that it took a monumental amount of time to set up the experiment, and we installed the shade nets really late. That's the point, okay? So the results I'm gonna show are these very late in the season. Okay, so within this context, I am gonna show you the results, and hopefully I can explain this. Well, this is just the models. The first thing to show you is that there was a lot of individual variation, so we had to run mixed models to account for the random effect of individuals. The green and the yellow are in the lights, ambient and drought, and the shade plots are blues and reds, okay? So what we see, and this is the weeks of shade, and this is the osmotic potential. So what we see is that the osmotic potential, as expected, decreased both in the ambient and in the drought plots. Notice that the drought start lower because we had actually exposed these plots to drought since last fall. Okay, so they're starting drier. What's interesting is that the osmotic, let me see what's happening here. Oh, these lines are not somehow the graph moved, okay? But so these were not. Okay? But the point here is that the osmotic potential in the light plots is decreasing, and it's not happening, so the osmotic potential is not decreasing as much when these plots are shaded, okay? So again, it seems that this shade, which presumably is accompanied by a decrease in non-structural carbohydrates, is not allowing these trees to osmotically regulate, okay? So there is a decrease of osmoregulation in the shade. I want to insist these are preliminary results because we still do not have the non-structural carbohydrates analyzed. And I also want to say that these effects are actually small, and this is a very artificial situation. We shaded these trees for 8 weeks. So, but what we need to do now is repeat this next year earlier in the season when these trees are not so stressed. So, and these results, so what I hope I've convinced you is that this non-structural carbohydrate storage, a large fraction of it seems to be really heavily involved in turgor maintenance and therefore in water relations. And this is consistent with models that suggest that there's large costs of osmoregulation in plants, okay? So I think we're getting close to the end, but this has implication on drought mortality. First, because it suggests that we need to pay more attention to pools of water. Until now, we've sort of focused on fluxes, on water potential, on cavitation. We need to pay attention to pools of water. And there is evidence that the depletion of non-structural carbohydrates, it was work by Mickey O'Brien in tropical seedlings, a very clever experimental design that he depleted seedlings and then compared them with enriched seedlings, and the depleted significantly died about 8 days earlier. In tropical system, that's a lot, okay? And this is another work that I did that Nuria did in Spain. I was collaborating, and she also grew these plants in the greenhouse. She measured different variables over time, and then she modeled mortality. And what she found very interesting is that, so I'm going to summarize these, is that she found that higher carbon assimilation under drought increased survival despite the cost. Okay, so survival seedlings or these plants survive more if they photosynthesize more, if they had higher stomatal conductance, meaning they were losing more water. And this survival was also related to soluble sugars and to the amount of leaf area, which is completely mind-boggling, right? You would say that having a lot of leaf would accelerate mortality. In this case, it delayed mortality. And that suggests, again, that these plants had no choice. Either we assimilate more carbon or we die for sure. So we're doing work now to try to understand the management implications of this in the context of fires. We have a lot of fires. There is no regeneration after very, very hot fires. So we need to plant seedlings. And how they do that is they grow these seedlings en masse, but then for one season, and then they store them in the dark in the freezer. And then they plant them. And of course, we suspect these seedlings are depleted in carbohydrates, and that actually is decreasing survival. So with this, I hope I have convinced you that plants, we knew that plants have abundant stored non-structural carbohydrates, and that a large fraction of these stored seems to be critical for plant-water relations. And this suggests, and there is evidence for this, that storage might be tightly regulated to prevent excessive depletion. And we've shown this in collaborative work with Megan Blumstein at Harvest. We have a recent paper showing a trade-off between storage and growth. And then with Henrik Hartmann's group in Germany, where they saw upregulation of storage under carbon depletion. And so with this, I want to thank you very much for the invitation again and for being here. Yeah, sure. Yeah, you're welcome. Thank you.
So we're staying in the field. We don't have ponderosa pine around here, but in the desert here we do have acacia trees, and Daphna Uni will present her work on acacia trees. She is from the Hebrew University of Jerusalem, from the Weizmann Institute of Science, and also from the Arava Center, the Dead Sea, Northern Arava Center.
Hello, my name is Daphna. I'm a PhD student. My work focusing on acacia trees and how they cope with extreme hot and dry environments. But today I will show you only one project from this PhD. So acacia trees are the iconic desert tree, keystone species in the desert, distributed all around the southern hemisphere in the most arid deserts of the world, the Namib Desert, the Sahara Desert, the Saudi Arabian Desert, and the most northern edge of this genus in the world is in the Arava and the Negev Desert of Israel, where we did long-term observatory research and wanted to understand the seasonality of photosynthesis and the activity of the trees. And we find out, surprisingly, that their peak photosynthesis occurs in the midday of the summertime. Also, as related to Anna's talk. So, but this is in the field, and you're welcome to read this paper. But we wanted to focus more on the physiological and especially on the stomatal regulation in terms of how water limitation regulate the stomata of acacia trees. So in the field, we have extremely deep and wide root system that we cannot control the amount of water that the trees are getting. So in order to answer our question, which was what are the physiological responses of acacia trees to limited soil water availability, we had to do a control experiment. So we focused on two species, Acacia tortilis, which is more rectangular shape, and Acacia radiana, which is more curly hair tree. And we did two treatments, control, and drought. I know drought is more long and complex than stop watering, so the system will demonstrate somehow the drought. And we wanted to understand how they respond to low water availability. So I added those marks so it will be easier to follow. So we collected seeds from the natural populations of the acacia in the Arava, germinated them by clipping to mimic the natural way they are germinating in the field. When they grew up a little bit, we transplant them in a unique pot system that differ between the plant, the soil, and the water. And we waited for 7 months. We wanted to do the experiments during the summer, and we put the saplings on a lysimeter system in the Hebrew University, belongs to Professor Menachem Moshelion lab. So this unique system measuring the weight, let's focus on one saplings to understand, measure the weight of the pot every 3 minutes. So those are 3 weeks of the experiment. Now we'll focus on 1 day. So you can see that during the night there is gradual irrigation. The pot reached to full irrigation, and then all the excess water drainage. And we want to reach to a plateau before dawn, before the sunrise. So this plateau is super important because after this plateau, the only change in the weight represent the daily transpiration of the plant. Only the water that transpired through the plant itself. But we didn't had only one. We had more than 50 saplings, and the second super important feature on this system is that all plants are subjected to the same drought. So based on their own transpiration rate, we can decrease the actual amount of water that we give to the plants. So here you can see also that we measure the atmospheric conditions, the radiation, and the temperature on VPD and relative humidity. Those are the plants in the two the treatments. And we also located probes in the soil, in the drought plants soil. We used the sandy soil, and you can see the decrease during the experiment. So now let's go to the results. Again, we have 2 species, 1 control, and drought. This is the midday canopy conductance, how much gram water transpired through gram plant in a minute. And you can see that both species decreased their stomatal conductance during drought. And now let's take in 75%. And also we measure the weight of the plants that represent the growth, the only biomass that gained in the plant. And if we'll focus on the drought period, you can see that we found some differences between the species. Acacia tortilis, the orange one, took his time with closing its stomatal, while Acacia radiana closed immediately, reduce its canopy conductance. In terms of growth, also, you can see that the slope of the Acacia radiana species decreased sharply, while Acacia tortilis remained similar to its control. And now, instead of looking at the whole experiment, it was 48 days during the summer. Let's focus on each one of those phases of the experiment, and we can see that the diurnal pattern of the canopy conductance during midday. So here you can see the time of the day and the canopy conductance, and here it's the total amount of water that transpired. So before drought, we see no differences between the groups. The 2 species reacted the same, had the same diurnal pattern. During drought, those species are graphs are the drought. So you can see 2 interesting things. First of all, the diurnal pattern of stomatal conductance, of canopy conductance, stayed the same in the drought. So the peak of canopy conductance again occurred during noontime. And the second thing is that Acacia tortilis, the orange one, had higher values of canopy conductance than Acacia radiana, and also higher variation, between the replicants. And we also recovered the plants after 2 weeks of drought, and we found that Acacia tortilis almost regained their canopy conductance to control, while Acacia radiana did not control it fully. So now, instead of looking at the time of day, let's take a look at the volumetric water content, the actual water that those saplings experience. So here is the volumetric water content, it's like percentage, and the canopy conductance. So in maximal volumetric water content, stomata and canopy conductance was high, and we were looking for the spot for the amount of water that the plants start to decrease their canopy conductance. And we found out that Acacia radiana, the green one, had close their stomata in higher volumetric water content compared to Acacia tortilis. And this gap allowed the Acacia tortilis plant to grow more in the period of low volumetric water content. Also in the recovery, we can see that Acacia tortilis in the first 24 hours reached a much higher, double the time of Acacia radiana canopy conductance. And also the slope of the recovery was higher. Short summary that we found that Acacia tortilis is closing its stomata slowly, have higher water use efficiency, and recover fast. So we'll call it the risky take species. And Acacia radiana is more cautious, have lower water use efficiency, fast stomatal closure, and now we are coming back to the field. As Anna also said, it's also my comfort zone, the field. So in the field, here you can see 3 case studies that we found out that in the main channel that represent in blue lines, the abundance of the cautious species, Acacia radiana, is much higher than in the secondary order channels. So the secondary order channels is characterized by fast movement of water, water are coming fast, going fast, in comparison to the main channel that drainage all the system and have higher available water. So it's nice that the physiological response we saw in the saplings in the lysimeter system also reflects the distribution of those species in the Vadi system. So let's sum up. Both species continued transpiring under low soil water availability, 5 to 9%, presenting a unique and non-trivial strategy of water spending. So why would a desert tree will transpire high amount and take a risky strategy? We think that the fact we did this experiment on seedlings, on saplings, young trees, represent their first life stage, and we know that the acacia trees invest everything they can in their roots in order to reach the water. So they prefer to suicide, or I'm reaching the deep soil layers water, or I prefer to not live in the desert. So to make it or to break its strategy. We also show that Acacia tortilis perform more opportunistic approach than Acacia radiana, and these results may explain their different topographic distribution within the Vadi system. So thank you for listening. Thank you, my supervisors and Professor Menachem Moshe Leon. Thank you.
One thing that people forget sometimes about drylands, that they are also a big source for fruit trees. One of them is almond, and Tamar we'll talk about if you want. and about photosynthesis.
Okay, so thank you, Tamir. Today I would like to talk about one of a very interesting project in my lab. We are seated in the Nevaeh Volcanic Center in the north, and I will be talking today on the wild almond, Prunus arabica, and the utilization of stems as a major source tissue for photosynthesis. So we were discussing this quite a lot during the last couple of days about the climate change, and within we have a lot of changes from increase in the CO2 level in the atmosphere, the increase in global temperature, and drought and the scarcity of water in different parts of the world. So with that, we have from the Food and the Agriculture Organization, we are aware of an increase in the world population. In the last 50 years, it's almost double. And it is predicted to increase and reach 11 billion people by the year of 2100. And this increase in population, together with with climate change, we will of course have an increase in the scarcity of food and world hunger. So almond, except of being one of my favorite food, we know this as a very big industry, and we are aware of the health and the food values of that exist in almonds. So the almond industry is a thriving, the almond field is a thriving industry, and it has been increasing around the world. We see an expansion, like that, thanks. We see an expansion of the area that we grow almond around the world, and the increased demand in the almond made the area of growing almond towards more drought and warm areas. And for example, California is the major producer of almond. More than 80% of the almond are being produced in California. And now the areas has expanded to, for example, the San Joaquin Valley in California, which is very dry and warm. In Israel, we have planting around the northern Negev, which is very dry and warm land. So, so during the development of agriculture, a lot of traits that have been that exist in wild almond species has disappeared during breeding for commercial cultivars. Now with climate change, there is a very important reason to look for these traits that will assist in breeding them into commercial cultivar in order to have almond trees that are more resilient to climate change, which increase their growth and, of course, yield. So we found a new wild almond species, the Prunus arabica, and recently it was also gathered into the collection of almonds that we have in Nevaeh. Currently, we have more than 150 cultivars of almond. And this species is very unique It was found around the mountain, the fertile mountain crust, which on one hand they have a lot of areas that are very arid and dry, and on the other hand, it is native to high altitudes with cold winters. So we see that it is native to a very extreme climate and growing conditions. So, okay, so one of the noticeable features of the Prunus arabica, as you can see, it does not look like the commercial cultivar, which is here the Prunus dulcis. This is Fahem. It's green stems. So this is a very unique trait. We could not see it in any other almond species. And these stems stay green all year long. And when we look at the commercial cultivars, the stems are becoming barked. So in order to further study this trait, we followed the surface area. We measured the surface area of the Prunus arabica, the wild almond, versus the Prunus dulcis, the commercial one. And we found that the stems, the green stems in the Prunus arabica are composed of 60% of the whole surface area above ground of the tree, while in the Prunus dulcis only 0.1% are green stems. And then, on the other hand, when we look at the Prunus dulcis, more than 93% of the surface area are green leaves, which are the major photosynthetic tissue. And in the Prunus arabica, it's around 32%. So, in the desert, especially the desert shrubs, stems are very common as a photosynthetic organ. And especially under very extreme conditions, like drought, and more even when the plant have no leaves. And then it becomes actually the major photosynthetic tissue when the plant have no leaf. And this feature kind of grab our attention and because it kind of reminds us what happens here in the Prunus arabica, the wild almond species, and therefore we decided to study this trait further. So in order to test what is happening with this stem and is it really have a photosynthetic capability, we first look at the stems of Prunus arabica and Prunus dulcis and made a scanning microscopy analysis. And we see that in the Prunus arabica, the stems on the epidermal layer are very rich in stomata, sunken stomata. And if we look at the second year of development, we see that they are still exist over there, very rich. You can see here the stomata per centimeter square. And in Prunus dulcis, on the other hand, first they start with a very low density of stomata on their stems, on their green stems when they emerge. But on the second year of development, they are covered with a thick barked layer. And then they are just disappeared. So we wanted to further get more into what happens within the stem, and we made cross-section of the Prunus arabica and Prunus dulcis. Here what we see is the second year of development, and we found a few distinctive features between these two species. First, as you saw, there are sunken stomata, but then they are open up to a very thick parenchyma layer that looks like palisade cells. So palisade cells are the tissue within the leaf that is the major photosynthetic tissue. They are very long and they are rich in chloroplasts. On the other hand, when we look at the Prunus dulcis, we see a peridermal layer. Peridermal layer is the one that is in charge for the bark formation, and we could not see peridermal layer in the at least as thick as that in the Prunus arabica. Another very interesting feature we found is that the Prunus arabica have additional 5 bundle bundle sheets, yes, sorry. And in the Prunus dulcis, we have only the main vessel. I can go more deep into that, but I think we have limited time. We further investigated the chlorophyll distribution, and thank you, and the level. We did extraction from stems from the Prunus arabica and Prunus dulcis. We see the first and second year. Basically, they have 3-fold higher chlorophyll in the Prunus arabica stems. And the distribution, when we look at the confocal autofluorescence under confocal microscopy, we see that it is located in the Prunus arabica just under the epidermis in the parenchyma tissue, while in the Prunus dulcis, it is distributed kind of all along the stems, starting from the chlorenchyma and into the phloem, xylem ray, until the pith. Next, they have stomata. Stomata are responsible for gas exchange and carbon gain, photosynthesis. So we wanted to check how these stomata respond to different environmental stimuli, and we did gas exchange on stems, and we checked the stomata respond on the stem for CO2. We see that the stomata respond to shifts in CO2 levels. We checked the response to light and dark transition. So under dark, the stomata close and respond to elevated temperature, and indeed also they close in response to elevated temperature, which further support their function under high temperature conditions, where they close the stomata and preserve water and increase their water use efficiency. So So we wanted to check how this stem photosynthesis function throughout the year. I will focus only here on the net photosynthesis. All this information also exists on a paper that we published. And we see that the Prunus arabica stem photosynthesis is very high. Actually, it reached around 85% of the leaf photosynthesis, so it's very high. And we see that it's functional throughout all year long. And what is very important is the winter time, when the tree has no leaves. So we see that we have another tissue source for photosynthesis and carbon gain. So as I mentioned, we have published 2 papers. One is which focus more on the physiological aspects. And another one on the mapping of this trait in the almond genome. And I want to thank for all the people that contributed to this work, in particular to Doron Holland and all his lab members, including Kamal Khatib, Tali Treinen, Hillel Bruckenthal, which is this is a part of also his PhD. Dr. Or Shapira, Ziv Atia, Shira Gal, Hani Tzemach, Edi Balsarov, and from the Volcanic Center at Bedagan, Dana Harouvi and Vili Ktwari. And short opportunity for me to share with you that we have granted by the Ministry of Innovation, Science and Technology with a big grant. We are looking for postdoc that will continue this very interesting work. So, thank you.
So wonderful work, but since we're talking about the possible exchange of it for commercial use, so how big is the fruit? How big is the growth overall of the plant?
Right, thank you. There's a lot of information in the paper, the mapping paper. We are actually in continue steps of getting this trait into commercial varieties. We already are in the second, in the F2 generation. We also received a very long breeding grant in order to get different traits, and this stem photosynthesis trait is also one of them, into commercial varieties. So hopefully in 10 years I'll be here again and I will show you the next commercial variety with stem photosynthesis. But we are on that. We have genetic markers. We know exactly how to screen it. And we have preliminary data that show an increase in yield in breeding populations from the Fachem together with the Prunus arabica, the F1 population. We screen them. We know that the ones that have stem photosynthesis also have an increase in yield. And this is only the first generation. So,
I have a question. So yesterday, Professor Kati said, she showed us, she showed us how she covered the stem and all the branches and make an experiment. Is this something you're trying now? Or?
No, we don't want to cover them. We like them as they are. Kathy talked about something that is also the more familiar word for it is corticular photosynthesis. It's also very popular to use this term. And it also supports different physiological aspects in the plant. Also, there are different publications on the resilience if you have the corticular or without the corticular. But this is two different strategies of the plant. We have barked plant, and corticular photosynthesis is for barked plants. So under the bark, there is some layer that have chlorophyll, and it actually can rescue the carbon that are coming from respiration and from other sources of the plant. There is a lot of CO2 within the plant. Okay, so if you give him some light, you can also get use of that. This is a different tissue that it's mimicking the leaf photosynthesis, and we want to kind of get something that the trees that are under harsh environment use it for resilience and to grow under very harsh conditions, but to get it into commercial varieties. So we are talking about getting only these traits without these small seeds. This is bitter, you cannot eat it. The Prunus arabica, it's like, if you want to die, this is the way. Yeah. But the idea is to take a trait or few traits. There are other traits. Also, this corticular, the bundle, the additional bundle sheets, we believe that they also, because there is photosynthesis in a very thick tissue, that you need to get all the photosynthesized the carbon that are from this thick tissue. The main vein is just too far away. So if it gets like under bundle sheets, so all the sugars, the carbon can get through it and get into the main vessel. Okay, so there are other traits here which are very interesting for us, and the idea is to make a super power almond tree. Yeah. Thank you.
So we get to our last speaker today. And for that, we are coming back to this place, to this campus. We have Ibrahim Salman, who studies here in the lab of Vered Sin, and Merav Saifan. Thank you. And the stage is yours. This will be very interesting and taking us somewhere different from what we saw so far.
A bit.
Okay.
A bit.
Please, Ibrahim.
So my voice is loud. You need a microphone. With the microphone is better. Okay. Thank you, Tamir. I will try to talk today to you about not only plants. There will be some insects, some pollinators, and I hope you will like it. You will all relate to the iconic image of a pollinator visiting a flower, and the visitation of a flower by the bee is to collect the nectar, the pollen, as a food. And mainly, in most cases, is a she. The bee is a she, which is a female foraging for food. And they collect the food not for themselves. They collect the food to feed others. In a way, they collect the nutrients, the lipids, the carbohydrates they found in the pollen to feed others. So the plants need these bees to visit them for a very simple reason, because they are very bad in having sex. So they need the bees, they need the pollinators for the room for sexual reproduction, okay? So the plants are anchored to the ground, they cannot move anywhere, and they still being attracted. They need someone to do the dirty work for them. But how this being done? It's a bit confusing, and as a simple, you think how it's being done. So there is several ways the pollinator going to the plant. And the attraction of the bee to the flowers is simply be either innate or learning preference based on adaptation or based on diet breath. So one of the ways is visual cues. And the visual cues can be different flower colors, different flower sizes. And these visual cues allow bees to discriminate between rewarding and non-rewarding species, individuals even within a species. However, these visual cues can also allow other insect groups to utilize other forms of information. It can be related to rewards, but not always. And since this is a dryland conference, and since my work here related to dryland and desert, this is not always the case. In most cases, if you're in the Mediterranean area, visual cues can be very, very important. But if you're in a desert, visual cues cannot be as good as other things. And why is that? Because when the bee needs to go to the outside, they are investing so much energy. It's very highly energetically costly. So when they want to go outside, they need to make sure they are doing the job. And visual cues is not enough. So what's the plant do? They're trying to invest in other source of energy, which is non-visual. They're sending chemical cues to the atmosphere as a immediate interaction with the others. So they're sending an information, and this information can be used as a way to communicate with the outside world. And this way, it allows the bees to use this information to locate the resources in the area. Okay? So the volatiles is when they've been produced mainly through the cytosol or maybe even through the mesophyll, they need to cross a very long way until they're produced. And what we used to know previously, that they've been emitted through the cytosol to cross to the plasma membrane, the cell wall, through the cuticle, and to the atmosphere. And we thought it's a kind of straightforward, just being produced and go away outside. However, this is not the case. It's a bit more complicated. There is a transporter proteins that takes them from usually cytosol, passing through the aqueous cell wall to the non-polar waxes in the cuticle, until they accumulate in certain numbers these chemicals in the cuticle, there would be another transporter protein take them from the cuticle to the atmosphere. So in this case, we have a cuticle that acting like a lock and key. So they're accumulating, they're emitting when they're when they're accumulating certain numbers. However, volatiles, like many other plant traits, they are changing over time. They are sensitive. They are varies in time and space. So in order to prove that, what we did recently, we collected the population from the desert. We were working on a desert plant called Matiola lividia, in Hebrew known as mantur. And what we did, we wanted to know if actually the plant can respond to stress. So what we did, we tried to stress the plant. We provided different stress, can be biotic and abiotic. Our biotic was whitefly infestation. Abiotic were in terms of irrigation manipulation. And I don't want to go into details of the experiment, but just the idea, what we found there is irrigation manipulation can actually provide changes to the volatile produced by the plant and whitefly infestation as well. Regardless now of the whitefly infestation, if we just focus on the irrigation manipulation, the main finding were there was an increased amount of esters, significantly increase in the low irrigation. And what is esters? Esters actually, it's a green leaf volatiles. It's a defensive volatiles. When the plant is under stress, they try to emit more of this. And this was proven, this was an indication for us when we tried to do behavioral bioassay in pollinator. The pollinator tried to avoid this, the plant that were producing more esters. Excellent, great. The story didn't finish here. This is a small scale variation. But biotic and abiotic variation is large. You can find biotic and abiotic variation everywhere. And this can lead to population that individuals that differ into through adaptation to different habitats. And what that's supposed to mean? That's supposed to mean that each biotic and abiotic variation in each habitat can lead to local adaptation as expressed in what? As expressed in individuals would have higher fitness in their home site. And home site means the population where they grow, okay? And one way is to measure local adaptation is through geographic distance. Geographic distance can be a good indication to measure the strength and the magnitude of local adaptation. So let me just walk you through what I mean here. If you have a gradient here of a different population from the desert to the north, taking all the population from the north, suddenly they are in the south, in the Midreshet Ben-Gurion. What you would expect to happen because we have a local adaptation? We would expect to reduce fitness, and this is mainly due to reducing gene flow and the environment become more differentiated. So it's very logical to expect, due to the local adaptation and due to this different population that grow in different regions, there would be floral signals also would be different. And because they are different and there would be reduced fitness, it will be less attractive, and possibly pollinator will prefer population that grow in that area. So in order to investigate what we did, we collected several seeds from different populations, and we took those seeds to Midreshet Ben-Gurion, and we grow them in a common garden experiment. And the common garden experiment just for them to have a common environment. And we provided a two irrigation regime with the low and with the high. And we measured the different variables, including physiology, size, primary sugars, volatiles, and pollinator observations. Okay, just want to share with you a very short video about us in the field. This is the 36 plots. We started doing the experiment when it was flowering. You can see Aviad doing the physiology work, me and Daniel doing the pollinator. You can see also two irrigation system. It was fun. Yeah. So starting with mainly with the results, as some of the stress indicators that we measured were stomatal conductance, and for stomata, it is an estimate to measure the estimation of gas exchange and transpiration through the leaf stoma. And what we mainly found that there is no actual differences between where you're taking the population, how much farther apart they are from the home site. It does not really matter. What really matters is the differences in irrigation. if you provided the better irrigation, there will be differences in they will open the stomata more. And then this is very well documented in literature because water stress usually erupt the photosynthetic rate due to stomatal closure, and it also prevent the inward of gas exchange. The other thing that I want to focus on, leaf temperature. And why is that? Because heat stress usually impacts the photosynthetic apparatus, and increasing leaf temperature has to do with reduced chlorophyll A fluorescence. And what we found here, that the farther you go from the home site, it's the hottest the plants were showing, the more stressed they were. And if you look also at the irrigation part, they were showing differences in leaf temperature. But the interesting thing that I would like to share is the southern population here were showing more sensitivity to irrigation. And this sharing the idea that it's not about the plant community itself, rather than the source of origin where the population originated. For flower size, similarly, the population that grow in the south showing small, tiny leaves, but the one from the north, they still contain their origin and they still produce larger leaves, larger flowers, sorry. And again, the sensitivity to the southern region were more pronounced to irrigation. We found that different sugars, several sugars were identified for Sinapis alba, the mustard. And the main one is, as always, we go to the sucrose. And sucrose usually cleaved to produce UDP, glucose, and fructose. I know it's too much information to share, but I would like to share information regarding starch, and this is the point that I want to make here. In Synapse, what we found, there's two types of starch. There's storage starch and transition starch. And the transition are usually stored inside the chloroplast during the day, and at night, they breaks down through maltodextrin to produce maltose. And maltose is a primary form of carbohydrate export. And what we found related to maltose, that they are emitting in a higher amount in the southern populations. The plant seems to be more happy. They have more sugars. And by increasing the distance, the farther apart you go from the home site, they reduce the amount of sugars. And the same thing for irrigation. There would be increasing amount of irrigation, increasing amount of sugars. For volatiles, just due to limitation in time, I just want to share that the major differences that we found in volatile production related to irrigation, I could not detect any differences in the source of origin on volatile production. It was mainly due to productivity. It's either high or low irrigation. Several volatiles being detected, mainly terpenoids and benzoates. But there's a very unique compound, it's andicane, which is an alkyne, is known to have a trade-off in function, either as a deterrent or as attracting to pollinators. I want to share you what we found regarding this compound because it was extremely interesting. The emission of this andicane increased significantly by distance. We've taken the one that farther apart the population to the Medraship and Gorion, they produced more andicane. It can be related to stress. It requires further investigation in order to know exactly what that's supposed to mean. For pollinators, and this is the unique thing about the research, we're now showing stress. We're now showing a different amount of volatile produced in different populations. But what would you expect regarding pollinators? So when we went to the field, we had some sort of predictions, but we were not expecting to find that the pollinators' visitation rate will prefer the one from the south. The dominant pollinator group that they were in the area, they prefer the one that they grow up. They didn't go to, in a way, their visitation was not significantly different from. They prefer the one from the south. As a summary of the results, if you imagine this is a gradient of your population from the arid to the Mediterranean, usually there is less stress here, more pollinator visitation, more stress in the northern population if you take them to the home site, which is in our case the desert site, and less pollinator. And we're assuming because the pollinators they prefer here, and this work still in progress. Flower size, sugars volatile increasing with productivity. The effects probably might be long-term. Even though northern population have more intense floral signals, pollinators still prefer the population they grow with. And by that, thanking my advisors, Shai, everyone who contributed, and also the bees, very important. Thanks.
I'm basically wondering what you show here is basically a trade-off between the ability of the plant to defend itself and the ability to call pollinators. And I'm wondering if the pollinators are generalists or are they specialized to these specific flowers? Because I would assume that if the pollinators are generalists, then it makes sense that as you go further away from home, you would like to invest more in defending yourself against herbivores or others, and less in calling for pollinators. And as you're closer to home and you know your enemies, you would invest more in pollinators. So what can you tell us about the specialization of pollinators?
The pollinators are the most ones that we identify are generalist pollinators. We couldn't detect specialist pollinators. The predictions that you're trying to make is very viable, very important, and I think we're kind of discussing the same points. But I want to make the point here. The visitation frequency to the northern population were little doesn't really mean it was not sufficient, okay? And we're now trying to do some analysis on the seed to the plant fitness data in order to actually be able to answer it. But your point is very viable, actually. Yeah. Thanks. Please.
Thank you, very interesting. The fact that sort of secondary metabolite production increases with productivity is sort of very general. Have you tried to standardize by biomass?
It's been standardized by mass, and we usually also. So there's a different way to transform the data for secondary metabolite, but what I did here was spimming, and spimming is you take in the flowers, you freeze them in liquid nitrogen, and you have a specific amount of flowers that been collecting for every single individuals from the field, and then after we doing certain procedure, we transform it based on the biomass. So we already know that it is standardized throughout all samples.
So we started with the carbohydrates, and we even finished with some sugars, so that we wrap up. It's very nice. I thank again all the speakers for today.
I use this opportunity to